students. So let's start with today's biology class. In the previous class, I have taken a live class in which we have discussed about the first process of the central dogma, that is DNA replication. Actually, it is not included in the central dogma, but the central dogma is the transfer of information from DNA to RNA and then to protein. So the first process was the DNA replication that we have discussed already, and I have told you how you have to attempt that question if it comes in your exam. Means what are the points that you should write, which diagram you have to make, and how much you have to write for the answer. Okay. So the next part that we are going to start in this chapter, that is molecular basis of inheritance, is the transcription process. So as you know, in the central dogma we have studied that the information is transferred from DNA to RNA and RNA to proteins, and that is in the form of code. Okay. So the first process of the process of copying the genetic material, genetic information from template or antisense strand of DNA into RNA is called transcription. What does it mean? So copying genetic information तो we have already discussed. कि यहाँ पे जो genetic information है वहाँ पे ये किसकी बात कर रहे हैं, क्योंकि ultimately we know that any information in the genetics is expressed in the form of proteins. तो proteins को बनाने के लिए पहले, जैसे you can say it is like a language. अगर हमें किसी को कोई information देनी है और हम एक code language में बात कर रहे हैं, तो that is a coding language. उसी तरह से जो ये information है, genetic information, इसको express करने के लिए protein बनाने पड़ते हैं, और उन प्रोटीन्स को फॉर्म करने के लिए इनको क्या करना पड़ता है, अपने कोडेड मैसेज को आगे फॉरवर्ड सेंड करना पड़ता है. तो वो डीएनए पहले आरएनए को सेंड करता है और फिर आरएनए जो है वो एक्चुअल में उस कोडेड इंफॉर्मेशन की हेल्प लेके प्रोटीन को सिंथेसाइज करता है बाय जॉइनिंग डिफरेंट अमाइनो एसिड्स. दैट विल बी इन द ट्रांसलेशन प्रोसेस, द नेक्स्ट प्रोसेस दैट वी आर गोइंग टू स्टडी. So here I have used a term template or antisense strand of DNA. So before starting with the actual process of transcription, we will first know some terms which we are going to use in this process. So first is the template or antisense strand. So the template strand is that strand of the DNA on which the RNA will be synthesized. Okay. Actually RNAs are of three types. When we will study translation, then I will tell you mRNA, tRNA and rRNA. So you know that we have already discussed about the types of RNA, and from this we will find that the mRNA will be synthesized over DNA, because you know mRNA is also in the form of a strand. जैसे DNA की strand होती है polynucleotide, वैसे mRNA की भी strand होती है, but that is made up of ribonucleic acid. Okay, that is why it is given mRNA name, because it is called messenger RNA. इसको messenger RNA क्यों कहा जाता है? Because it transfers the information from DNA to synthesize a specific type of protein. Okay. So here you can see the strand of the DNA which will act as a means on which the RNA will be synthesized is called as template strand. Okay. So here I have shown the two strands of DNA, because you know DNA is a double-stranded structure. Out of the two, one will act as a template strand on which the mRNA will be synthesized, which is the process of transcription. The other name of template strand is called antisense strand. Why it is called antisense strand? It is very important point, because sometimes students may get confused why it is called a sense strand. Sense means you know किसी भी चीज की अगर हम बात करते हैं कि कोई अगर हम एक स्पेसिफिक वर्ड के लिए बोल रहे हैं तो उस वर्ड की सेंस बन रही है या नहीं, मीनिंग है या नहीं उस सेंटेंस का, दैट इज कॉल्ड सेंस. The word same sense is used here. Okay, but why this strand is called antisense strand, though it is the strand on which mRNA will be synthesized? Okay, let me tell you why it is called antisense. Because you know, for example, if I am just giving an example, this is the DNA strand. Okay, and it has a specific sequence over it. Like sequence क्या होता है कि जो nitrogenous bases होते हैं, base pairs होते हैं, उनका एक specific manner में present होना, that is called its sequence. So because I will use this term sequence again and again, that's why I am telling you, you should know what does this mean. कि sequence का क्या मतलब है. For example, I have a strand of DNA example, I have taken this strand, तो यहाँ पे क्या था A, T, C, G, T, A, C, G. This is one of the strand of DNA. So इसकी जो complementary strand होगी, उसपे क्या sequence होगा T, क्योंकि DNA में क्या होता है T, not U. A pair up with T, T pair up with A, C with G, G with C, T with A with T, this again G and then C. So can you compare these two strands with each other? सिर्फ T और U को आपको इग्नोर करना है because in the mRNA there is no T, but in DNA there is no U. So they are equivalent to each other. ठीक है, तो आप इन दोनों strands को compare करिए. Are they both same? Yes, because they both are complementary to each other, and this is the actual sequence of mRNA which will decide the type of protein synthesized by the cell. Is it clear? तो इससे ही डिसाइड होगा आगे कौन सा प्रोटीन फॉर्म होगा बाय द प्रोसेस ऑफ ट्रांसलेशन. ठीक है, नाउ इज इट क्लियर व्हाई दिस स्ट्रैंड इज कॉल्ड एंटीसेंस? क्योंकि दिस स्ट्रैंड इज द एक्चुअल सीक्वेंस ऑन द बेसिस ऑफ व्हिच द प्रोटीन विल बी सिंथेसाइज्ड. सो दैट इज व्हाई दिस स्ट्रैंड इज कॉल्ड सेंस स्ट्रैंड, क्योंकि उसका कोई यूज नहीं है. वो स्ट्रैंड सिर्फ एमआरएनए को बनाने में उसने हेल्प किया, बट एक्चुअल में जो सीक्वेंस दूसरे स्ट्रैंड के ऊपर प्रेजेंट होगा वही डिसाइड करेगा कि प्रोटीन कौन सा फॉर्म होगा. दैट इज व्हाई दिस स्ट्रैंड इज कॉल्ड एंटीसेंस, बिकॉज इट विल नॉट डिसाइड द टाइप ऑफ प्रोटीन सिंथेसाइज्ड, एंड दिस स्ट्रैंड इज कॉल्ड सेंस स्ट्रैंड बिकॉज इट विल डिसाइड द टाइप ऑफ प्रोटीन फॉर्म. नाउ दैट इज व्हाई इट इज कॉल्ड नॉन कोडिंग स्ट्रैंड एंड दिस इज कॉल्ड कोडिंग स्ट्रैंड. मीन्स नॉन कोडिंग बिकॉज इट विल नॉट सिंथेसाइज द प्रोटीन्स, बिकॉज मैंने आपको बताया था कि प्रोटीन्स को सिंथेसाइज करने के लिए एक ट्रिप्लेट कोड होता है जिसमें 3 बेस पेयर का सीक्वेंस होता है. ठीक है, जब हम ट्रांसलेशन स्टडी करेंगे देन यू विल फाइंड इट. So that is why this strand is called sense strand or coding strand. I hope this point is clear. This is very important. So the next term in this that we will discuss before starting the process is transcription unit. So transcription unit is the segment of DNA that takes part in transcription, because you know the DNA, full DNA does not undergo transcription because you know that DNA is very long. It is around 2 meters and it can accommodate in the cell only by coiling. ठीक है, that we have already discussed in the coiling process of DNA. So if you will see that DNA is so long, so it is not possible to transcribe the full DNA. Only a few, you know that over DNA there are genes present, because we have already discussed that a small unit of DNA is called gene. तो जो different genes present होती हैं इसके ऊपर, that is actually the main part of the DNA which will undergo transcription. Okay, because you know transcription is basically for the formation of mRNA which will further synthesize the proteins, and these genes can express themselves only by synthesizing the proteins. Okay, they can express themselves only by synthesizing the proteins. So for that we require only this much part which has a specific code for the protein synthesis of a specific protein. ठीक है, so this is clear that the whole DNA will not undergo transcription, only a specific portion of the gene itself. पूरा जीन भी अगर हम देखें तो एक जीन के ऊपर काफी सारे जीन्स होते हैं. When we will study operon system, then you will see it that there are many genes over a specific part of DNA. Not all the genes will be undergo transcription. So they have a specific segment of DNA where the transcription actually takes place. This is called transcription unit. Here you can see I have shown a transcription unit. These are the 2 strands of DNA, one in the 3 end to 5 end and other is in the 5 end to 3 end, that is in the anti-parallel direction. So this side here you can see is the promoter site. Promoter means किसी भी चीज का start होना. So this is actually the initiation site or the promoter site from where the transcription actually starts, and this side is the terminator site where the transcription will stop. Okay. And this gene which is present in between is called the structural gene, which actually has that code of formation of a specific protein. ठीक है, तो अब एक DNA के सेगमेंट के ऊपर भी जीन होते हैं, जीन के अंदर भी फर्दर जीन्स होते हैं, but only the structural gene will be synthesized, will be transcribed other than other part of the DNA. So is it clear? This is the transcription unit and it has promoter site where the transcription will start with a specific codon. जब हम इसका पूरा डिटेल प्रोसेस स्टडी करेंगे then you will find and which codon is present over terminator site, which is a specific codon which will start and stop the transcription process. Okay, so now we will start with the transcription process in detail. So let's see the process of transcription. Actual process starts from here. Transcription process. That was just for your knowledge. If the question comes, you have to start from the definition and then process. So first step in the process of transcription is activation of ribonucleotides. As we have discussed in case of DNA replication, the first step was activation of deoxyribonucleotides because there the DNA strand has to be synthesized. But here we want to make the strand of RNA, so we will require ribonucleotides. Okay. So first step will be the activation of ribonucleotides. You know that the inactive form of either ribonucleotides or deoxyribonucleotides, it is in the form of monophosphate. Okay, and they has to be converted into triphosphates, which is the activated form of ribonucleotides. So here you can see there are 4 types of ribonucleotides in the inactive form, which are present in the nucleoplasm. As the deoxyribonucleotides are present in cytoplasm, but this is present in the nucleoplasm because the deoxyribonucleotides, when the process of DNA replication takes place, they have to transfer from cytoplasm to nucleus. But here it is not like that. They are already present in the nucleoplasm. They just have to be activated. Okay, so they get activated by converting into triphosphates. So there are 4 types of ribonucleotides: GMP, guanosine monophosphate; CMP, cytidine monophosphate; AMP, adenosine monophosphate; and UMP, uracil monophosphate instead of TMP, which is present in DNA. ठीक है, तो T की जगह यहाँ पे क्या होगा? Uracil. So these are the inactive forms present in nucleoplasm. Now they will be converted into triphosphates: GTP, CTP, ATP, and UTP. These are the active ribonucleotides, and it will take place in the presence of phosphorylase enzyme. Okay, so the enzyme is phosphorylase. This is the first step in transcription. Okay, so if you have to write in the same way, there is no need to write in the form of lines. Try to write in a flow chart की form में आप उसको लिखने की कोशिश कीजिए. So you will write like this: in the presence of phosphorylase, they will get activated. Next step is the initiation. Initiation, as I told you, in the transcription unit, there was promoter site and termination site. So promoter site is the site where the actual transcription starts. So you know that in case of DNA replication, the main enzyme was DNA polymerase, and this is the transcription process. The main enzyme is RNA polymerase. Okay. तो इस पे भी अगर कॉम्पिटिटिव एग्जाम्स वगैरह में क्वेश्चन आ जाता है, the main enzyme in RNA transcription is RNA polymerase, and for DNA replication is DNA polymerase. So initiation will start at the promoter site. So initiation takes place at the promoter site of transcription unit. Here I have shown the DNA helix with the antiparallel strands. See, this is the transcription unit. This is complete transcription unit. I have shown the helical form. This is the promoter site. For example, at this site, the initiation of RNA transcription will start. So this site is called promoter site. And I told you that this site has a specific sequence. क्योंकि हमें पता कैसे चलेगा कि DNA के ऊपर कौन सा साइट एक promoter site की तरह वर्क कर रहा है. तो जो एक ट्रांसक्रिप्शन यूनिट होता है, अगर उसके आप promoter site के सीक्वेंस को देखेंगे, then you will find this is TATAATG. Generally जो यही सीक्वेंस उसके ऊपर प्रेजेंट होता है, which is present at the promoter site at which the initiation of transcription takes place. So here you can see its name is Tata. So it is also called Tata box. इसको क्या बोलते हैं? Tata box. इस टाइप के क्वेश्चन आपके कॉम्पिटिशन वगैरह में पूछे जा सकते हैं. That is just for your knowledge. Why it is called Tata box? Because they have a sequence of TATATA. Okay, that is right. Okay. So here you can see I have shown the promoter site, and this is the RNA polymerase. this is the RNA polymerase enzyme, and here I have shown a small dark structure which is called sigma factor. क्योंकि जो RNA polymerase है वो कुछ सब यूनिट से बना होता है. RNA polymerase is an enzyme. तो कोई भी enzyme जो है वो प्रोटीन से बने होते हैं. प्रोटीन के भी उसके अंदर क्या होते हैं, फर्दर पार्ट्स होते हैं which are called sub units. So the RNA polymerase enzyme has some sub units and it is present in the inactive form, but when the RNA transcription starts, it takes place, then it gets activated. Okay, तो ये एक्टिवेट कैसे होगा, जब इसके ऊपर सिग्मा फैक्टर अटैच हो जाएगा. तभी मैंने यहाँ पे RNA polymerase के साथ यहाँ पे डार्क स्ट्रक्चर शो किया है which is showing the sigma factor. So this RNA polymerase will attach itself to the promoter site and this will starts the process of transcription. The next step we will see, and it always takes place in 5' to 3' direction. So here this is the 3' end and it will take place from 5 to 3 end. It's on 5 to 3 end, not the DNA 5 to 3 end. अब 3 end पे attach करेगा तो ये 5 end होगा इसका opposite, so it will take place in 5 to 3 end direction. Next step is elongation. So let's see the next step. Okay, so next step in transcription process is called elongation. As you know, the initiation of mRNA synthesis was started. Next step is the elongation, because you know elongation is to stretch something. When the base pairs or the activated ribonucleotides, they will attach to each other as well as with the DNA strand, then they will form a chain-like structure which we require. Okay? So we have two steps in this: base pairing and chain formation. Base pairing is the complementary ribonucleotide base pairs with the deoxyribonucleotides. So here I have shown this step, that is elongation. इसमें क्या होगा कि जो एक्टिवेटेड राइबोन्यूक्लियोटाइड्स थे, जो कि इनिशिएशन या फर्स्ट स्टेप में हमने स्टडी किया था कि एक्टिवेटेड राइबोन्यूक्लियोटाइड्स वर सिंथेसाइज्ड, दे विल बेस पेयर देमसेल्व्स विद द कॉम्प्लीमेंट्री डीएनए स्ट्रैंड, विद द टेम्पलेट स्ट्रैंड ऑफ द डीएनए. जैसे यहाँ पे क्या था कि ये डीएनए की टेम्पलेट स्ट्रैंड थी और एंटीसेंस स्ट्रैंड थी एंड दिस वाज द सेंस स्ट्रैंड. यहाँ पे इनिशिएशन स्टार्ट हुई. इलोंगेशन के टाइम पे क्या होगा कि जो आरएनए के बेस पेयर्स हैं ये इस तरह से यहाँ पे आते जाएंगे और अपने आप को डीएनए के साथ कॉम्प्लीमेंट्री सीक्वेंस में अटैच करते जाएंगे. ओके, सो दिस विल बी कॉल्ड एज बेस पेयरिंग. सो here I have shown the reaction or the process like GTP, CTP, UTP or ATP. They were the activated ribonucleotides. When they base pair with the DNA, the sequence or the nitrogenous bases of the DNA or deoxyribonucleotides, they will be converted into monophosphates with the release of phosphate plus energy. क्योंकि उसको activated हमने इसीलिए किया था कि वो मोनो से ट्राई में कन्वर्ट हो जाए, because you know when the phosphate bond is synthesized, then energy is stored in it. When the bond is break, then energy will be released. So here you can see the activated triphosphates, when they will pair up with the DNA complementary strand, then they will be converted into monophosphate with the release of phosphate and energy, and this energy will be used to form the bond between RNA nucleotides, ribonucleotides with the deoxyribonucleotides. Clear? So this is called base pairing. Next step is the chain formation. अब जैसे मैंने आपको बताया कि यहाँ पे ribonucleotides अपने आप को अटैच करते जा रहे हैं इस तरह से, but there is no bonding or linkage between these ribonucleotides. तो इनको आपस में भी तो अटैच होना पड़ेगा ना इस तरह से. So this formation of bond between the adjacent ribonucleotides will be called as chain formation. This will be also done in the presence of RNA polymerase. So here I have shown the elongation process, both the steps. This is the DNA helix. तो जहाँ पे भी ट्रांसक्रिप्शन होती है, ट्रांसक्रिप्शन जो यूनिट होता है, वहाँ पे वो सब प्रोटीन्स, जो सभी एंजाइम्स जो कि टोपोआइसोमरेज, हेलिकेज और सिंगल स्ट्रैंड बाइंडिंग प्रोटीन, तीनों स्टेप्स, तीनों एंजाइम्स हमने जो डीएनए रेप्लिकेशन में स्टडी किए थे, वो तीनों भी यहाँ पे यूज होते हैं. जहाँ पे भी डीएनए को अनबाइंडिंग करनी है, सिंगल स्ट्रैंड में कन्वर्ट होना है, so these three enzymes will always be there: helicase, topoisomerase and single strand binding protein. So they will attach themselves here. This helix will be open and the RNA transcript here, this is the chain of RNA, is synthesized. When this elongation process will start, after initiation the sigma factor will be released, क्योंकि इसने एक बार RNA polymerase को एक्टिवेट करना होता है, उसके बाद जैसे एक बार RNA synthesis स्टार्ट हो जाता है फिर ये sigma factor हट जाता है. Next step is the termination. As you know, the transcription unit has a terminator site. When the RNA polymerase reaches the terminator site, it will come to know that now they have to stop the transcription. क्योंकि उतने ही पोर्शन का उनको स्ट्रक्चरल जीन का उनको ट्रांसक्रिप्शन करना था. जैसे ही आरएनए पॉलीमरेज टर्मिनेटर पे आएगा तो क्या होगा, आरएनए पॉलीमरेज अपने आप को सेपरेट कर लेगा और ये जो हमें आरएनए की चेन मिलेगी इसको हम क्या बोलते हैं, प्राइमरी ट्रांसक्रिप्ट. क्योंकि इसके अंदर फिर फर्दर चेंजेस होंगे, थोड़ा बहुत मॉडिफिकेशन होगी, एक्स्ट्रा जो न्यूक्लियोटाइड्स है, वेस्ट न्यूक्लियोटाइड्स वगैरह वो सब रिमूव करेंगे. तो ये प्राइमरी ट्रांसक्रिप्ट है, नेसेंट जिसको हम बोलते हैं, नया जो बिल्कुल फॉर्म होता है. This is called primary transcript of RNA. Later on, it will be undergo modification for further process of translation. Okay, so this is the RNA transcript. And the next, that is the final step, is duplex formation. तो जो DNA अपने आप को उसने सेपरेट किया था, अनवाइंड किया था, वो सारा फिर से अपने आप को हेलिक्स फॉर्मेशन में सिंथेसाइज कर लेगा, so that it can takes place its original position. So this is all about the process of RNA transcription, and you have to write these points in the same sequence. क्योंकि आपको किसी भी बुक में नहीं मिलेंगे, you have to write in the same manner and the same points should be mentioned when you will write the answer. Okay, here is the diagram. First step was the initiation that I have shown in the previous video, in the previous step, and elongation and termination. This diagram also you have to draw with all the points explained. Okay, this is all about transcription.
The genetic material is stored in the form of DNA in most organisms. In humans, the nucleus of each cell contains over 3 × 10⁹ base pairs of DNA, distributed over 23 pairs of chromosomes. Each cell has 2 copies of this genetic material. This collectively is known as the human genome. The human genome contains about 30,000 genes, each of which codes for certain proteins. Although thousands of genes in the human DNA code for proteins, large stretches of DNA in the human genome are transcribed, but they do not code for proteins. These regions are known as introns, and they make up about 95% of the genome. Some of this non-coding DNA controls gene expression, but the purpose of much of it is not yet understood. This is a fascinating subject that is certain to advance rapidly over the next few years. So we know that all the information of the human genome is coded in the DNA in genes in the form of nucleotide base pairs. How this coded information is used to produce functional products like proteins is known as gene expression, and it essentially consists of 2 important steps. The first involves copying of the coded information in DNA to a RNA molecule, which takes place inside the nucleus of the cell, and this is known as transcription. This RNA molecule then leaves the nucleus, and the information of this RNA molecule is used to produce functional gene products like proteins, and this step is known as translation. In this video, we will take a brief look on the process. First, the enzyme RNA polymerase forms a pre-messenger RNA molecule. This happens in the nucleus by using base pairs complementary to the base sequence of the DNA template strand. In the second part, the pre-mRNA is edited by more enzymes to produce a desired mRNA, which is the final product of the transcription. So let's look at the first section of this process, which is the formation of the pre-messenger RNA, which essentially takes place in 3 steps: initiation, elongation, and termination. Now the main enzyme that is involved in this process of transcription is the RNA polymerase. The RNA polymerase has the main job of copying the information of the DNA to a new RNA molecule. RNA polymerase builds the new strand from the 5' to the 3' direction, adding new nucleotides to the 3' end. So the first step in the formation of a pre-messenger RNA molecule is the initiation, in which the enzyme RNA polymerase binds to the promoter region of the DNA. Now let's suppose this is a gene that is shown in the green that needs transcription. At the beginning of each gene, there is a sequence of base pairs known as the promoter region of the gene. The promoter region serves as a guide which tells the enzyme RNA polymerase where to attach. Once the RNA polymerase binds to the promoter region, it separates these 2 strands of DNA, providing the single-stranded template needed for transcription. Only one of these strands is used in the process of transcription, which is known as the template strand. This strand always runs from the 3' to the 5' direction. This is because of the fact that we just discussed. That is, that RNA polymerase can only synthesize the new strand from the 5' to 3' direction. So it can only use a strand of the DNA running from the 3' to 5' direction as a template, as the new strand that is copied is always copied in the anti-parallel direction. So after the RNA polymerase binds to the promoter region of the DNA, the second step in this process is elongation, which is the process of copying the bases on the DNA template strand to form the new complementary RNA strand. The RNA polymerase uses nucleotides according to the base pair complementary rules to create a RNA copy. In the previous videos, we learned how the DNA is copied by DNA polymerase to form an identical new strand of DNA. In that case, the DNA polymerase added the base thymine when the template strand had the base adenine, and it added the base cytosine when the template strand showed the base guanine, and vice versa. But in case of transcription, the RNA molecule that is produced contains the base uracil instead of thymine. So there are no T's in the base pair sequence of RNA. Whenever the RNA polymerase detects the base adenine in the template strand, it adds uracil in the forming RNA strand. So this is how the enzyme RNA polymerase adds new base pairs in the 3' end of the growing RNA chain, and this is known as elongation. The next step is termination. So just like we had a sequence of gene known as the promoter region which signaled the enzyme where to start, we have regions of the gene that basically signal the enzyme where to stop. These signals are known as terminators, and the signal that the RNA transcript is now complete. Once the RNA polymerase reads and transcribes the terminator regions, the newly formed RNA transcript is released from the transcription complex. So this was the first section of this process in which the DNA is used to produce a RNA molecule. This RNA is known as the pre-mRNA or the pre-messenger RNA. The prefix is used here to show that this mRNA is not yet ready to leave the nucleus and start the process of translation. So the next section of this process of transcription deals with how this pre-mRNA is converted to a mature RNA molecule. Imagine that you want to publish a new book and you have printed